Assalamualaikum and a very good morning. So here is our presentation for project SECB2103 Bioinformatics 1, which is Find a Gene. So our group have already chosen a target protein, which is tumor protein P53, which is also called as TP53. We are from group 6, section 01. So my name is Dasni Barubiti Haja and my other group mates are Nurin Fatini binti Zulkefli, Sarveswari anak perempuan Wasudevan, Yasmini Abiramavalli Tinaharan. So now let's move on to our first part, which is protein identification and why we chose TP53. So as you can see in the slide, our target protein is tumor protein P53, which is also known as TP53. This particular protein, famously known as the guardian of the genome, due to its vital role as a tumor suppressor. So we actually chose the human variant, which have the accession number as shown in the slide, and you can see the reasons why we have chosen the TP53 protein. So here the first reason is that it is well annotated RefSec entry. This particular protein have a well annotated RefSec entry, which is making our project to be reliable at the starting point. So, and the second reason is that TP53 actually belongs to an unseen and highly conserved gene family, because it is also present in both the vertebrates and invertebrates. So then our search is actually more focused to invertebrate genomes, because we have uncovered that the invertebrate genomes are seems to be very less. Let's move on to the next slide, which is BLAST search method. So as for this step, we utilize T-BLAST-N, which allows us to search translated nucleotide databases using our protein query. So our protein query sequence was the human TP53 protein, and we searched against the TSA database. So to specifically target under studied organism, we applied a filter, taxonomic filter for Annelida, and Annelida is segmented worms. We did this because we wanted to choose for a lower annotation level to set the stage for novel discoveries. So now let's move on to the next slide, which is BLAST search result. So we successfully identified multiple significant hits across this various Annelida species. So here you all can see the sequence identity of these hits as the picture shown here. It is kind of range from 41% to 75%. So then this particular range indicates a clear but divergent homology, which means it is a good novelty candidate for us. So now let's move on to the next slide, which is selected alignment. So from our previous BLAST search results, we selected the specific alignment like you all can see in the picture in the slide. So then the alignment metrics are highly robust. The score of 213 bits and an incredibly low e-value as shown in the slide is kind of mathematically, it could guarantee that this is a true evolutionary homologue of the TP53. It can be said as it's not a random match. So now then you all can see the identity here, which is 45%, and the positives is 61% with gaps 6%. So now I will explain about why we chose this particular sequence alignment. So here I've stated the reason, because this sequence is strong enough to confirm that the real evolutionary relationship to TP53 is exists. So when they have a very strong confirmation, it can be it can give us a strong it can be our strong candidate for novelty in the further steps.
Next, I will present the successfully isolated and translated our target sequence from the marine polycystic worm, that is Phagocypio elegans, that as shown here, they have 216 amino acid partial sequence that was reconstructed from a specific transcription shotgun assembly transcript. It also contains essential core DNA binding domain characteristic of a P53 large tumor suppressor. So our next step was to provide that this sequence is completely unique by testing the novelty of this sequence. Next, we need to verify the sequence novelty by running a reverse plus TP search against a global non-redundant protein database. The results show that our top match isn't non-Phagocypio elegans protein, but rather a hypothetical protein from a completely different species, that is Regia species. They share a 70% sequence identity with an e-value of 78216 because there is no identical and pre-existing match for the Phagocypio elegans P53 protein in the database. We can officially confirm that this sequence is entirely novel. So we can proceed to the next part. Next, from the result, we choose 5 sequence for the conservation analysis later. First one is hypothetical protein. Next, second one, tumor protein P73. Next, tumor protein 63. The fourth one is DGYRCD as 11540. And the last one is DGYRCD as 11541.
So once we got our 5 homologous protein sequences, we are going to find the results for the multiple sequence alignment. How we're going to do it? So we're going to put all our protein sequences in the EMBL EBI muzzle tool. So once we put in the tool, we'll get this result that's been shown here. When we look at the alignment, three things we can understand about our new protein is that it has a core active part. So the middle alignments here, they have long rows of this star. So this means the core part of the new sequence is almost perfectly identical to the other proteins. And the second one is it belongs to the worm family. So our new protein sequence, right, it matches with the other marine worms such as Lamelbracia satsuma. So this proves our new protein sequence is correctly classified as part of worm group, which is annelid. The final one is it's a partial piece. So when you look at this, there is a lot of dashes at the very beginning and the very end of our sequence. So this is simply because our sequence is a short fragment from a database while others are full-length proteins.
So moving on to the evolutionary analysis of our target protein. So we constructed a phylogenetic tree to determine exactly where our novel Phagocypio elegans sequence fit within the wider annelid lineage. So for our methodology, the multiple sequence alignment we generated earlier was used as the input, and the final phylogenetic reconstruction itself utilized a neighbor joining algorithm processed via the FastMe pipeline. And to ensure our tree layout wasn't thrown off by noisy data, the sequences were first cleaned and then optimized using BMGE to filter out any hypervariable regions, giving us a highly reliable topological structure. So, if we look closely at the visual output, the tree successfully resolves 3 key evolutionary trends. So first, you can look at the tight terminal node containing Lamelbracia satsuma and Regia pisacea So this satisfies the established biological taxonomy. Both of these organisms are deep-sea hydrothermal vent tube worms, which belongs to the family of Cyboglindiae, and these short horizontal branch lengths represent a high sequence conservation and a very recent common ancestor. Second, the tree shows a clear internal control for gene duplication. At the bottom, you can see the two sequences from Dimorphalus gyrociliatus splitting cleanly into their own distinct sister pairs. So this confirms the pipeline sensitivity is in distinguishing the paralogous P53 variants within a single organism's genome. And finally, to address our target sequence placement. So the novel Pygospio elegans P53 partial sequence branches out at a distinct node immediately ancestral to the main polychaethlate. So while our primary BLAST B screen shows that it has strong sequence identity with Regia pisacea, this tree reveals its true evolutionary trajectory and it forms its own unique line of descent right from the core group. So, to conclude, the distant base tree securely validates that the biological entity of our novel sequence, confirming its authentic placement within the ancestral P53 tumor suppressor superfamily networks of marine annelids. Thank you.